So I think the slides are visible basically, and in between when I ask the question, like please try to respond and give the answer accordingly. You can like unmute yourself or directly talk the answer and all, or else you can directly like comment the chat box and all. If or if you have any doubt, you can also feel free to ask in between. So yeah, I think I will start. So today we will be starting with the V4 section of the uh, V4 section of the NPTEL course Cell Culture Technology. So I will just move on to the first question itself. So for the first question, which of the following items cannot be autoclaved? Polystyrene, polycarbonate, stainless steel, or borosilicate glass? Which might be the answer? You can type in the chat box, no issue. Okay, so the answer to that question is polystyrene. So I will just give a brief brief explanation of some of the items that are used in autoclave. I will also move into some of the explanation for autoclave, but I will start with the autoclave bags. uh, Sorry, I will start with the autoclave then. So I think that will give you an idea. You know autoclave what is, but I just giving an explanation for that. So basically, autoclave is a machine that is used to provide an elevated temperature and pressure as compared to ambient condition. So basically, you are putting any kind of material there that is not easily decomposed at high temperature and high pressure. And if you are using a cell culture, any kind of media, any kind of glass beaker, or any kind of like plastic falcon tubes, or any kind of whatever you want to autoclave, you are putting it in that particular instrument. Then the instrument actually elevates the temperature as well as the pressure within that particular autoclave, so that under that condition, under that stress condition, the microorganisms like bacteria, virus, spores, etc., or any other kind of contaminants gets kind of eliminated at that particular stage. And then you are after the autoclave process has taken place, you remove that particular what you say, like particular media or any kind of stuff you have put in the autoclave. Then you are using it for particular culture, because the afterwards, once the autoclave has been done, the materials that were present within the autoclave is kind of sterilized and all. So you can basically use some kind these decontaminated materials to incubate whatever cell culture you are needed, right? So because in order to prevent contamination, we need the sterilized stuff inside, right? So that's the main role of an autoclave there, because it provides that elevated temperature as well as pressure, so that the materials that you are require is getting sterilized there. And basically it uses this combination of heat, pressure as well as time which ensures the sterilization process. And this is a type of dry heat and as compared to other dry heat or chemical sterilization method, this kind of sterilization method is actually like much more effective as compared to that in this case. So basically the heat and pressure are produced from the water when the steam that is generated from the water that is present within the autoclave, because there's actually, if you move on to the components that are present within an autoclave, you can see that this is the autoclave. This is an image of the autoclave here. You can see that there are several components there. First one is the pressure chamber. You can see that this is the pressure chamber, right? So mainly it is made up of stainless steel to withstand the high temperature and pressure that is happening within the autoclave once it is on, right? So basically it acts as a chamber to enclose all the steam as well as the pressure that is generated there. So we are actually pouring some water into the autoclave, so there is a specific region so that you can pour the water inside it. And whenever you are doing an autoclave, you have to check whether the water level is proper or not, because without the essential amount of water within the autoclave, the steam won't be generated, and you won't get that adequate sterilization at that time. So water level has to be maintained, and there's a lid or door there. So once it has been maintained there, you are putting in the substances like whatever material you need to autoclave inside an autoclave bag. Then you are properly sealing the autoclave bag there, and then you are putting it up to a particular level. Basically it can accompany each and every autoclave can accompany a particular amount of material inside it. So once that have been achieved, you put the autoclave bag inside, then you seal the autoclave using a lid or steel. You can see that there are several nuts and bolts there. You tighten it up because in order to maintain that high temperature and pressure there, you have to correctly tight it up in between so that the pressure and steam is maintained there inside. And then there is a meter there also, right? The pressure gauge. So it measures how much pressure it has been released there. Basically about in each and every autoclave there's a particular temperature or a pressure that has to be reached there so that the adequate sterilization takes place. So you are basically sealing it up, and after sealing it up you are switching it on, and once it's switched on, there is steam generator there, right? Yeah, as I said, there is water inside the autoclave. Once the steam generator is on, it starts to make steam out of the water, and this steam in turn produces the pressure because it is a sealed environment, right? Within that autoclave, it is a sealed environment. So at that time, there is a steam generator there. The steam in turn elevates the temperature there, and also it produces pressure within the autoclave, and this leads to the sterilization process there. What sterilization take place in that case? What sterilization process take place in that case, or wet sterilization? And and also the as it is at a high pressure, it promotes that sterilization environment there. And also apart from that, there is a wastewater cooler there. So if water has to be removed from the autoclave directly, then it cools down the water so that whenever the water moves through the pipes that are present in the autoclave, it won't hamper those pipes and all. So once the sterilization has completely taken place, you actually switch off the autoclave, leave it for like 10 to 15 minutes, then you leave the open the valve there. There's actually a steam release valve there. You can see that in this case. It can be at the top or at the bottom, it depends. So once it is released, the steam actually goes out, and the pressure comes down as well as the temperature comes down, and afterwards you just open it up, keep it for open for some time so that the excess steam gets out of the autoclave, and then you take it out, take whatever the materials out after the proper sterilization and all. So these are the basic components of an autoclave. And also coming to the bags that are used in. So I have said that before you are switching on an autoclave, you have to keep the materials there. It's not like you directly keep the materials there. Basically, you put it in a bag called as autoclave bag, and then afterwards, once the autoclave bag has been put and it has been tightly sealed, then using any rubber band or any kind of sticky materials, whatever it is, and then you are put the autoclave bag into the autoclave, and then do a wet sterilization. You switch it on so that it can do the sterilization for that particular time. So basically, if you look into the materials that make up a autoclave bag, it is mainly made up of polypropylene, which is a type of plastic, and it is quite durable and can also withstand temperature up to 135 degrees Celsius during the autoclave bag, because it is usually at 121 degrees Celsius, right? For about 30 to 50 minutes you are carrying out the autoclave and all. At 15 psi or 15 bar pressure you are giving. So basically that have to be there, and it should be thin because it should not be hard and all. Like it should not be thick. It should not be that much thick, as well as it should not be that much thin. About approximately 50 micrometer of thickness should be there, and so that it could have that particular strength, and it should be resistant to tearing and all, right? So because it actually providing a sterilization at a high temperature and pressure, so it should be maintained there. And many are transparent, so that you can identify a company. Some are not transparent. It depends on the company you are buying the autoclave bag from, and it comes in various size and colors. Basically, you need that in order to handle the biohazardous material and all. There is a bag that comes under different color. Black color bags are there if you want to do auto-radiography stuff and all, radioactive material if you are handling and all. So it comes under different colors and size, and that depends on what we are autoclaving, whether it is a waste or whether it is a media or anything else. And also there are also temperature indicators that are present within the bag that can be attached to a bag, so that you can know that the temperature has been achieved. so far, whether the temperature has come or not, so that it gives an indication of whether autoclave is actually happening or not. So yeah, we will also discuss one of the questions according to this particular option also. So yeah, so this is one of the basic stuff that we use when we are doing autoclaving and all. So anyone has any doubt? Yeah, so I think it's quite understandable. So coming on to the next question. So question number 2, what helps in changing the color of an autoclave tape? First option is pressure, second is time, steam or glass. As I said, right, like we use autoclave bags, some of them have some temperature indicators and all. This is the question like that is based on that. So what would be the answer? You can type in the comment box, no issue in the case. Right or wrong, it doesn't matter. Okay, so the answer to that question is steam, because pressure is produced there, but what pressure is produced by steam that is produced generated there by steam generator, right, from the water. So time is not an indicative why autoclave tape changes color. The actual reason is because of the steam that is generated within the autoclave. So basically, these autoclave tapes are basically used in autoclaving. Right? Its name suggests basically that, and basically to indicate if the specific temperature for sterilization has reached. So basically, in an autoclave, we have to provide this much temperature, 121 degrees Celsius, and the temperature is 15 psi or 15 bar, and you are basically keeping that for about 30 to 50 minutes within an autoclave until this temperature and pressure has released, as reached, so that the sterilization is adequate there basically in this case. So basically these autoclave tapes are present in the like together with the autoclave bags, or you can just purchase it additionally and then paste it up in the autoclave bags and all. So once this temperature has been reached, the autoclave tape actually changes its color. So usually if you look into the color, you can see that it is actually beige or blue in color initially. Autoclave tape beige is actually like a yellow brownish color. So whenever it is heated up, this particular color changes from that. The beige or blue color actually changes to a black color. So it indicates that only at a particular temperature this color is achieved, that this color transition actually takes place. And this color transition actually gives you an idea that the autoclave temperature is getting reached and or the autoclave is happening properly. It's an indicative of that temperature basically. So basically if you look into the composition of this autoclave tape and all, it is basically made up of lead carbonate. So lead carbonate actually gets in the presence of steam or at a high temperature, in the presence of steam which is at high temperature, it gets converted into lead oxide. So that's actually giving you the color, the black color there. So that is indicative of the whether the temperature has been reached properly or not. And it is this lead carbonate is protected by a resin or a polymer, so that it gets easily degraded or decomposed to show that particular change. So this is why autoclave tapes are used in case of autoclaving and all. And the color change, that the transition from the beige or blue to black color is due to the presence of the steam that is generated due to steam generation that is taking place within the autoclave, right, upon time. So does anyone has any doubt regarding this? I think it's quite clear.
So one, you can check the temperature and the pressure in the pressure gauge,
Yeah,
right?
pressure gauge you can check.
So, and so like you put this tape on the items before placing it in the autoclave. So then how can you figure out how can you see
No,
the
like
color change and
inside the autoclave you cannot check. You have to take the materials out. Sometimes what happens is that if the meter is you are using is right is faulty or not. Sometimes it might get faulty. If the instrument is faulty or not, you cannot check whether the autoclave is proper or not. So whatever happens when you are using a autoclave tape, you have to take out the sample after autoclaving and see whether that color changes happened or not, because sometimes if any fault or any other like malfunction happen within the meter, you cannot you will think that sterilization has actually happened, but it might not have happened at night. So this is like a secondary indicator. It's not like a primary indicator. It's secondary indicator that temperature has or steam generation has properly taken place or not.
Okay, sir. Thank you.
Okay, so I think that question is clear. I will just move on to the next question then. So question number 3, this autoclave tape dash sterility and indicates that the proper dash was reached during the process. Which would be the answer? Okay, so the answer to this question is that the autoclave tapes does not assure sterility and indicates that proper temperature was reached during the process. So basically, I have said given you a small explanation about the autoclave tape. Right? Basically, it only indicates whether the temperature has been reached or the steam generation was proper or not. It does not assure you sterility and all. But whenever you are packing that material into autoclave, that will give you how that like particular how well you are packed the material into the autoclave and how much time it was taken for that particular sterilization to take place, and also how you are using the material once you are doing a sterile culture, right? So that all assures sterility and all the autoclave tests. These are just indicative of the whether the steam generation was proper and where adequate temperature has came up at that particular autoclave process or not. So this is the basically like from the previous explanation that I gave is pro that you get the idea about this answer. So anyone has any doubt regarding this? It's quite basic. Basically, if you have heard the previous explanation, you can basically answer this question, and it's basic. So I will just move on to the next question. So coming on to question number 4. The objective for which microscope is placed below the stage and which one is placed above the stage? Which might be the answer? First one is the upright microscope, inverted microscope. Second is the upright microscope again. Next is the inverted microscope or upright microscope, and the fourth one is the inverted microscope and or the inverted microscope. What do you think might be the answer to this question? Okay, so I will just give you the answer. So the answer to the question is inverted microscope, upright microscope. That is, the objective for the inverted microscope is placed below the stage and the upright microscope is placed above the stage. So I will just give you an explanation of like what you all know this basic stuff. So basically, a microscope is an instrument that is used to provide an enlarged image of small objects, right? If you are looking into a specific slide, if you consist of a water droplet layer, if you are looking into a microscope, you can see several microorganisms that are swimming through the water droplet, right? So it is basically used to enlarge whatever is present within that microscopic environment there. So you can get an enlarged image of all those like substances or any other kind of microorganisms that are present there. And it depends on the microscope that you are using, whatever your application is, whether it's a fluorescence microscopy, whether it's a confocal microscopy, whether it's just a light microscopy, you need to analyze a particular sample. It depends on that, right? You need to say blood sample, you need to see what all kind of blood cells are present there. So what you need to analyze the morphology of xylem or phloem or any other kind, whatever it is, like you need a microscope to visualize that, right? So basically, one of the common familiar type is light microscope or an optical microscope, right? Like high advanced one-light compound microscope or any other like electron microscope that is newly discovered one is there basically. So basically it's used for all these kinds of convenience for all these kinds of examination as well as analysis in this case. So if you look into this is the basically I am just giving a diagram basically of whatever a microscope holds, right? So basically there's an ocular lens or an eyepiece. So basically this is the region where you put your eyes and you look into the microscope, and the sample is kept there, right? So that's why the name actually take place. It's called as eyepiece there, right? And there is a dipolar adjustment there. Basically within underneath the eyepiece and all, you can see that there is a dial there. You can adjust the dial so that the eyepiece moves in and out, so that you can view comfortably, or you can adjust the magnification of the image that is taking place. And also there's a nose piece which consists of all these objective lens, right? So the objective lens actually consists of different type of lens which contributes to different magnification. There will be 10 time magnification, 40 times, 60 times, 100 times, and basically if the magnification increases, then chances are that you can look into much higher magnified area, or much smaller object that can be looked into a 10 time objective lens, right? In some cases you can also have to use a 10 time objective lens in some cases. And there's an arm there basically which holds all these components to the particular components that are present beneath the objective lens and all. And you can see that there's a stage clip as well as an aperture there. So basically you put the sample there, and then you adjust the magnification there, and then you look into eyepiece, then adjust the size using a coarse adjustment as well as a fine adjustment there, right? So coarse adjustment basically, like if you want to move the microscope rapidly means not that rapidly, but up to a certain point, so that you can easily like you can get a clear image on the eyepiece. So fine adjustment is much lesser, employs much lesser movement as compared to a coarse adjustment, so that fine details can be magnified or changed there. And there's also stage control. You can move this particular stage clip, right? When you are placing a like sample or any cover slip or any kind of any whatever it is, you can basically rotate this out so that it can move like to and fro movement will be there. You can also move right and left, like it can move towards you or away from you basically if you are adjusting it. And then there is a diaphragm, so illuminator is there which provides the light, which is obviously the light source. So once you are switch it on, the light will be provided there, then condenser will condense the particular light source onto the cover slip, onto the area where you are viewing the, or you are putting the analysis for. And also there is a diaphragm there. Diaphragm actually condenses the amount of light, or controls the amount of light that is entering into the specimen, so that it can be viewed whether it is bright or dark, it depends basically. And there is a light switch as well as you can there's a brightness adjustment there. So basically this is a basic components of a microscope. It might seem that there are several components, but if you know how to properly utilize a microscope, you can basically like how to use a microscope, you can basically, it will be easy for you in this case. And basically we do that practicals and all, right? So it's basically an easy one. So this is the components of that constitute a good microscope, and this is basically a compound microscope that is shown here. And coming on to the question, basically you are saying that the objective for the inverted microscope is placed below stage, and upright for the upright microscope is placed above stage, right? So basically these are the two types of microscope, basically upright as well as inverted microscope. So in case of inverted microscope, you can see that the objective lens is what looks at the specimen, right, the sample, right? So light actually comes from the top, then the sample is kept there, sample stage is there, and then the light actually passes through the sample, then it moves into objective, and you can actually visualize it in the eyepiece. So basically if the sample that you are using, or the specimen that you are made, the slide you are made is actually transparent, then you can use an inverted microscope. So if it is not transparent, then you have to use an upright microscope, because the light might come from this region, then it reflects back, so that you can actually go through the objective lens, you can actually retrace the path here, right? And then it moves into the eyepiece, because if the sample slide, or the slide that you are using, or any kind of substrate that you are using for to keep the sample is actually opaque or translucent in nature, then you have to use an upright image, because if you are using an inverted microscope, so the lamp is actually at the top, right? So basically it comes down, then it will block that stage, because the specimen or the sample is actually opaque in nature. It cannot pass the light through the objective. So in case of like upright microscope, that's not the issue. It can easily pass the light, and then you can easily watch the whatever the sample is through the eyepiece, or you can directly attach it to a camera. Several of these microscope actually provide a provision so that it can be attached to the camera and record whatever you are seeing there. So if you want to have a live recording and all, you can usually use a microscope having this live recording camera that can be attached, and then later record it out, and whatever purpose you are using, you can basically use it out. So basically these are the two different type of upright as well as upright as well as inverted microscope. So I hope this answer is clear. So objective for the inverted microscope is placed below stage, and this upright microscope is placed above the stage. So anyone has any doubt? Okay, so I will move on to the next question then. I think it's clear. So question number 5. The source of the light and condenser for dash is which microscope is placed below stage, and which microscope is placed above stage? The answer is quite clear from the previous image that I have shown. So anyone has any answer? Okay, I got an option 1. Any other answer? Yeah, so you're right. So the option number 1 is right answer. The source for the light and condenser for upright microscope is placed below the stage. You can see that in the upright microscope, right? The source of light, that is a condenser, you can see is placed below the stage. As well as in case of inverter microscope, it is placed above the stage. You can see that the source of light is placed there. I'm not going to give a detail because I think I have properly explained it in the previous slides. So I will go into the next question. Question number 6. What parameter is important to consider while buying a lens based on if you want to use plastic or glass, like whatever like material you are used for spotting the sample and all, whatever it is, there is a specific recommendation of microscope, because if it is plastic, then some specific recommendation is requirement is needed. In case of glass, it is specific different recommendation needed. So what will be the option? First one is of focal length, magnification, numerical aperture, and density of light. Yeah, I'm getting option number 3. Any other option? Numerical aperture I am getting.
2.
Magnification, okay. Any other option? Okay, so the answer to the question is numerical aperture. Answer to the question is numerical aperture. So basically, I think magnification is dependent, but what sample you are visualizing, basically, right? So if you are looking into plastic or glass, numerical aperture matters, because it is something that depends on the refractive index of the substance, right? Because for plastic, the refractive index will be different as compared to that of glass, right? So magnification will be present, but if you are if you want to get a clear image, you have to take a microscope where the lens should have a particular numerical aperture there. So that dictates how clear the image will be. Once the image is clear, then you can slightly change the or tweak the magnification here and there, so that you get a clear image and all. So I think the answer to this question is numerical aperture. So basically, I will give a. So I think that's quite clear, right? So if you have any doubt regarding that, magnification is important, but when you are comparing between the different types of nature of the materials that you are using, right? So I think numerical aperture is needed. So if you look into a microscope lens, there is something written as n is equal to 1.4, 1.2 or 1, like something like that. So that's what numerical aperture means. So numerical aperture is a crucial parameter that quantifies the ability of a objective lens to gather light and resolve the fine specimen based on the fixed object distance that those fine specimen within the sample actually possess. So this is just a mathematical equation. Numerical aperture is equal to n sine theta. As I said, it depends on the refractive index of that particular medium between the lens and the specimen, right? So the specimen is actually kept in either a plastic or a glass. So if you know the refractive index, then basically you can actually add up to that refractive index. Then you can actually visualize the substance that are present within the particular sample, right? So basically that's why we are using numerical aperture there. That's why the factor numerical aperture is dependent there, because it depends on the refractive index of the medium, because this question is specifically asked based on the refractive index of the plastic or the glass there. And basically you can see that like there's a lens there, there's a sample is basically there, and you can see that the theta is the half angle of the maximum cone of light that can enter or exit the lens, right? And then you can visualize light is gathered at the eyepiece after the objective has gathered light out of it, and then you are visualizing it yourself, right? So this is the equation. Basically numerical aperture is equal to n sine theta basically, and n is the based on the refractive index that is present in the objective lens as well as the specimen, that is the component that you are using for keeping the sample there. So basically the answer to the question is numerical aperture. It is used for considering while buying a lens based on if you want to use plastic or a glass there. So I hope this answer is clear. Anyone has any doubt? And also, like, coming on to some of the important other aspects of the numerical aperture, a higher numerical aperture indicates a greater ability to collect light. So more the numerical aperture, right, more is the ability of the lens to gather that light, so which will give you much more brighter or contrast image and all. So it also enhances the clarity of the image, because what you need is the clarity, right? So more the light gathering power, more will be the detailing of the image that you are getting, and more will be the clarity there. So that's one aspect of the importance why we are looking into numerical aperture. And next is the resolution. Basically, if you look into like sample and all, you can see that if you look into two different points that are close to each other, then a good microscope should have the ability to resolve those 2 points at that specific distance itself. Sometimes if it is not clear, then chances are that it will get blurred, and chances are that you will see the 2 points together. But if the resolving power or the numerical aperture is higher, chances are that the microscope has the ability to resolve these two points together as it is with the specific distance that is present within them. So higher numerical aperture value allow the microscope to distinguish between smaller details in the specimen. And also medium dependence, because as I said, it depends on the refractive index, right? The equation is actually called a numerical aperture is equal to n sine theta, which is the theta is the half angle of the cone of light that is coming out of the specimen. So based on the refractive index, if we look into the air has a approximately refractive index. If you look into higher magnification rate and all, right, so then you need to add a immersion oil. So immersion oil also has a higher refractive index, so it's add up to the refractive index of air there, making it up to a 1.5 watt, so that the resolution of the microscope is increased there. And it also depends on the angular aperture. Basically, as the angle which the light objective is there, the maximum angle at which the light can enter the objective. So basically leads to as the angle increases, you can see that the n sine theta, right? So as angle increases, then you can see that the sine theta value move from 0 to 1, right? So it intends increase the numerical aperture there. So this is another importance of a numerical aperture, why we prefer when we are buying a microscope there. So I think this is clear. Anyone has any doubt? Okay, so I will move on to the next slide then. So moving on to question number 7. Which of these following is not a colligative property of a material? Which might be the answer. First option is osmolarity, color of solution, depression in freezing point, and elevation in boiling point. Which might be the answer. Okay, I am getting option number 2. Yeah, so the answer to the question is the color of the solution. Basically, all the other properties, right? Osmolarity, dispersion in freezing point, elevation in boiling point, these are all colligative properties. So what are colligative properties? So colligative properties are properties that of a solution, is not of a solid or anything. It's a property of a solution that depends on the that produce some characteristics which is dependent on the number of solutes that are present in that solvent molecule. So basically, it's the ratio. It depends on the ratio of the number of solute particle to a number of solvent particle, not on the nature of the chemical species. Just it depends on the number of solute to the number of solvent particles that are present within that particular solution. So if you look into a type of colligative property, so first one is the vapor pressure lowering. So the presence of solute reduces the number of solvent molecules at the surface. So basically, sometimes the solute mixes together and sometimes it does not. So based on the when you are adding a solute into a solvent mixture, the homogeneity of that particular solvent mixture will lower, right? So this leads to a decrease in vapor pressure of the solvent because the components that are present within that particular solvent mixture, when you add a solute from the outside, actually changes the composition of that solvent mixture solution itself, right? So based on the uneven distribution or the uneven concentration of the solute to a solvent mixture, there are chances that the vapor pressure of the solvent might get decreased in this case. So if you compare it with a homogeneous solution to that of the solution that you are just prepared by adding the solute molecule from the outside, then chances are that this will show a much higher vapor pressure. The homogeneous solution will show a much higher pressure as compared to the next one that you are just made out. So in this case, so the vapor pressure decreases if you are adding a number of solute molecules into that particular solvent mixture. And also boiling point elevation is there in this case because you are adding something into that component, right? So chances are there will be other interactions that are taking place in this case also, and it also increases that particular boiling point within that particular solvent solution that you have just made. So it increases the boiling point elevation. It elevates the boiling point in that particular non-homogeneous solution, and the freezing point actually gets dropped down because the homogeneity is getting affected there. So this is another property of that particular colligative property because chances are that when you are adding a solute mixture into that particular solvent, right, there's a depression or decrease in freezing point there, and osmotic pressure is also getting changed there because if you are having a homogeneous solution, then chances are that it will move through a semi-permeable membrane easily. But in case of that is a osmotic. So that's a different thing. So osmotic pressure is getting changed, right? Because the homogeneous solute that are present within that particular solution will exert a pressure there. But when you are adding a solute out of this, then the pressure chances are that this pressure gets changed accordingly. So that actually in turn affects the movement of this particular solute molecule across a semi-permeable membrane from one solution to another solution. So those are kind of colligative properties that are actually useful when you are making a cell culture on a buffer or any kind of system like that. So I hope this question is clear. Anyone has any doubt? Okay, so I think this is clear. So I will just move on to the next question. So question number 8. Choose the most acidic solution based on its pH. Option 1 is compound A pH 1, compound B pH 5, and compound C pH 8, compound D pH 14. Which might be the answer? Okay, I am getting option number 4. Compound D pH is equal to 14. Any other answer? Yeah, so the answer to the question, you are right. Option number D, pH is equal to 14. Okay. So I will just give you a brief explanation. So if you look into the pH, this value is rarely used, right? It's not that much commonly used as compared to this term pH. So pH is the negative logarithm. You can see the equation here. pH is equal to minus log concentration of H plus, right? So it's a negative logarithm of the hydrogen ion concentration in solution. So basically the hydrogen ion is concentration is the thing that dictates like what a pH is, what the whether the solution that you are like examining is acidic, either acidic or it is basic in nature or it is neutral in between in nature. So the scale can actually start from 0 to 14, right? Not from 1, 0 to 14 in this case. So if you look into a neutral solution, it have a pH of standard pH of about 7, right? So if you look into a pH less than 7, if it is 0 to 7, 0 to 7 like that range, then it is considered as an acidic solution, right? If it is from 7 to 14, then it is considered as a basic solution there, right? So based on the scale that you are getting, based on the scale that is based on the concentration of hydroxide ion that are present there, then the pH can be of a buffer system or a culture system can be determined there, and the measurement is actually taken with the help of a pH meter, right? So it consists of an electrode there. So basically you are dipping that electrode into a solution there. From that you are getting a reading. Some of the pH meters are basic. You just need to dip it into a solution. Some of the pH meter you have to check the temperature first, then you have to calibrate, then you have to check the pH. So that all depends basically. So if you look into a pH, then it's kind of an inverse basically. So it is a negative logarithm of hydroxide ion, that's the base ions, right? OH minus is something that determines the basic condition of that solution, right? So if you look into a scale, it's similar, it starts from 0 to 14, right? 7 is the neutral pH. So from 0 to 7 is considered as the basic condition. In case of a pH, if it is 0 to 7, it consists of it is considered as an acidic range. But if it is pOH is between 0 to 7, it is considered as a basic range, right? So from 7 to 40, instead of considered as a basic in case of a basic condition in case of pH, pOH is considered as the acidic range there. So you can see that the equation is similar to that of pH, just this particular term OH is replaced by OH minus. So pOH is negative logarithm of concentration of hydroxide ions there. So at 25 degrees Celsius, if you look into values, if you calculate both of them, it turns out to be 14 in this case. So does anyone has any doubt regarding this? So basically it's the inverse of pH that you are basically observing, right? So there's not much difference. You just need to distinguish the terms here and there, and at which pH range which one is acidic and which one is basic, and neutral is quite common in both. Is common in both of them. So I think it's quite clear. I think it's quite clear. Explanation is also quite clear. Does anyone have any doubt? Okay, so I think I will move on to the next question then. It's quite clear basically, like if you know the basic chemistry and all, you can basically answer that. So question number 9, clinostat uses to cancel the effect of gravity and is used to stimulate zero gravity conditions, which is the probable factor that a clinostat uses here, refractive index, solubility in a liquid, rotation, or a UV light. So anyone knows the answer? Clinostat basically. It's not that a kind of a term that you might have like come by, like is not used that much. Okay, solubility in a liquid I am getting, refractive index I am getting. Any other answer? Any other answer apart from that? Okay, so the answer to that question is rotation, the first option. So anyways, so basically it is a device or a rotation, it is not commonly used. So it is uses rotation to cancel the effects of gravity. Like it's actually kind of a propeller where you are taking at one end of the propeller you are keeping sample, in other one you can keep sample, or you can adjust the weight accordingly. Then in between it is connected to a motor there. So when the motor is switched on and when the samples are placed, it actually rotates at a specific RPM there. So it's similar to that of centrifuge there basically in this case. So when you are using a clinostat there, right? So when it is rotating, when at a particular RPM and all, it nullifies out the gravity effects within that particular sample that is actually rotating along the clinostat there. Basically, you can see that in case of outer space research and all, you can see that in ISS, right, International Space Station, they carry out all these experiments using this clinostat and all. And also, if you look into astronaut training and all, right, when you are training an astronaut, you are putting it into a chamber which consists of all this pressure taken out and all, so that it stimulates the space there and all. So it is also similar to that. Clinostat also similar uses this kind of techniques and all. So basically, if you look into space station and all, it rotates, right, actually within the space it actually rotates to produce that anti-gravity effects and all, and also sometimes it rotates to produce that gravity effects and all. So in this case, clinostat is similar to that. When it rotates, actually it cancels out the effect of gravity, and then it simulates zero gravity effect. So it provides a microgravity environment there. So basically you can study a particular cell culture or a particular cell culture that is under your observation, how it will proliferate or divide, how it will behave in this clinostat, that in the presence of this microgravity environment there. So basically this is actually a research that is currently taking place in case of cell culture embryo of spider web, how in the presence of a clinostat how spiders will behave and make their spider webs and all, whether it will be similar to that in a normal environment or whether it will be different. So basically it is advantages of a clinostat is that it can provide that microgravity simulation and all. So allowing the cell to study the cellular responses and all in vivo conditions, and also enhance cell growth. Basically in clinostat, which is an instrument, which is basically a clinostat there. And it can culture the cell in 3D, three-dimensional spheroids and all can be cultured in this case of clinostat, in the presence of this clinostat. And also how physiological relevant that particular culture will be if you are growing a hypostatic spheroid, how it will affect the metabolism within that particular culture, and also the structural properties of how it will affect the structural properties of the spherical cell and all. So that clinostat is used there basically. So this is kind of a research that has been going on, kind of rare, but it's still going on, how microgravity can affect the cell culture characteristics or the phenotypes there when it is grown there. So anyone has any doubt? Microorganisms do grow in zero gravity basically, but it doesn't mean that you are removing all the air and all the culture that are provided there, right? So it does grow, but the phenotype will be different from that when it is growing in a gravity given environment. So yeah, that's the answer basically. You are providing it the air and all. It's not like you are removing all the air and all, everything, the pressure will be there, but the only thing is that the gravity is getting reduced there. It's not completely eliminated or something, but it's getting reduced up to, that's why it's called as a microgravity environment there. Anyone has any other doubt? Okay, so I think this is quite clear. I will move on to the next question. So coming on to question number 10. In a T75 flask, what does the 75 stand for? For option number 1, it has a space for 75 cells. It was made in 1975. It has a culture volume of 75 millimeter square available for culture. It has a surface area of 75 centimeter square available for culture. Which is the answer? T75 culture flask is actually a culture flask used for cell culture and all. Some of you might have actually seen it. I am getting option number 4. Any other option? Okay, again I am getting option number 4. Yeah, so you're right. So the answer to the question is it has a surface area of 75 centimeter square available for cell culture. So basically you can see that this is a cell culture flask, right? So basically it's made up of plastic. But the difference is that based on the cell culture or the type of cell culture or the amount of cell culture you are growing, you can actually talk to a vendor and then buy this flask, right? So sometimes it will be smaller, sometimes it will be larger, or it can in between. So basically based on the size and the volume size or the growth area that you are providing in a centimeter square, or the volume that you are incubating within the culture flask, the type of culture flask that you are using might change. So there are four common types of this culture flask. First one is the T75, sorry T25, so for that T75, T175 as well as T225. So the T stands for is followed by a number that indicates that is the 25 indicates the surface area in square centimeter. So that's why it asks the T75. What does it mean? It means that it has a surface area of 75 centimeter square available for culture. So a T75 flask, most probably this one, has a growth area of about 25 centimeter square and is used for smaller scale culture application. Sometimes you need only a smaller amount of cell culture to carry out whatever the experiment you need to carry out, right? And it can employ about 7 ml to 10 ml of that culture media or the culture incubator culture, whatever it is. T75 offers a growth area of about 75 centimeter square, and it is commonly used for, it is a standard cell culture. Basically the question itself asks T75, right? So it is a standard cell culture flask that has been used here, this one, and it holds a working volume about 15 milliliter to 25 milliliter. And T175, it has a larger growth area of about 175 centimeter square, used for high culture volume about 35 to 50 ml culture and all. So and T225, sorry, T225, which has a growth area of about T25 centimeter square, and larger cell culture can hold maximum cell culture quantity in this case about like 45 ml to 70 ml culture. So these are what the term dictates actually. So there are the four kinds of flask: T25, T75, T175 as well as T225. And depending on how much culture you want to do, the requirement actually changes, and you can change accordingly to that. So anyone has any doubt? Okay, could you please repeat that question basically? Like I haven't completely read that. So animal cell culture is used basically. Okay, okay. So I think in plant cell culture, I think is different, right? Basically, if you look into, I am not that much experience in that, but whatever I have done in my UG and all, if you are doing a plant cell culture, you actually using a kind of a falcon tube, right? Rather than doing this cultures kind of cell culture, this kind of T25, T75 fast kind of, we are actually putting a particular falcon tube there, right, up to a particular size, right? So and afterwards, when you are pouring your culture media, basically made up of agar, then you are tilting it down so that it makes a slant there. Then you are putting up the culture or explant. What you say? It's called an explant, right? So explant, you are taking it out, a sterilized one, and then you are in sterile environment. You are putting it up, and then you are covering it with a cotton plug so that air can enter more easily and out of it without getting contamination and all, right? So then you are putting it in an incubator for some time. So I think like in plant cell culture, we are not using this particular kind of cell culture type. It's specifically used for animal cell culture. In case of plant cell culture, basically you are using a falcon or a falcon type, what is a boiling tube, a boiling glass tube or something like that, and then you are culturing it inside that particular, then you are covering it with a cotton plug, right? So I think it's specifically used for animal cell culture. So I think that's clear. But do check it out also. Like I think it's specifically used for animal cell culture only. Okay, I think it's clear. So I will just move on to the next question. Anyone has any other doubt? Okay, so I think it's clear. So coming on to question number 11. So the which lens in a compound microscope produce the first image dash the microscope, like objective or outside, eyepiece or in outside, objective or inside, or eyepiece or inside, which might be the answer. Yeah, I'm getting option number 3. Any other option? I'm again getting option number 3. Yeah, so the answer to the question is you are right. First one is the objective lens in a compound microscope produces the first image in the microscope, right? So because the objective lens is the lens that is close to the sample, right? The slide or the sample, whatever you are looking at. Eyepiece is close to your eyes because the image produced by the objective is analyzed by the eyepiece to produce the final image, right? So in a common microscope, the objective lens is responsible for producing the first image, which is real and inverted. So consider this area first. So this is the objective lens. So this is the object there. So consider that this the objective is like when you are like doing a optical diagram and all, right? Lens diagram and all. There's specific like instruction that you follow. So your object should be like this. It should be pointed upwards like an arrow, or pointed downwards like an arrow, and the light should move within the arrow point itself. So when the light actually comes out from the objective, this is a convex lens, right? The objective is a convex lens. So if it moves through the middle of the convex lens, then it will not converge. It moves in a straight line. So whenever it hits the side, then chances are that it will get converged up to a certain point. It diverts or diverges itself from that native path that should travel into another line. Then it then points in a, then these two rays of light, right? It actually points out a certain point. So it produces an image there. So this is the image point where the first image is produced by the objective lens. So you can see that if the objective is like this, then the final image produced by the objective where the ray of light actually converges is actually inverted, and it is a real image. It's actually the real image, kind of the like what you say is such as it is that converges into a point, right? So whenever an image converges into a point, or light rays convert image into a point to form an image, is called as a real image there. And then afterwards eyepiece actually analyzes this image, right? So whenever if you look into directly into eyepiece lens, you can see that it actually diverges itself into an infinity. So then you have to cross the path actually. So there are certain instructions that it's sometimes it is difficult to understand, sometimes it is easy to understand. So if you actually try to extrapolate or try to converge the 2 points that are produced by the eyepiece, you can see that the image will actually be enlarged. This is the final image. This is the first object image. This is the final image. You can see that it's much larger as compared to that of the image that is produced by the objective lens. So it actually magnifies out the image that is produced by the objective lens, the eyepiece. So it's actually produce a virtual image, because if you draw a line from here to here, you can see that it never converges. It actually diverges away from each other towards moves towards infinity. So those kind of images are called as virtual images. So it gives a so the objective lens actually produces a real as well as a magnified inverted image of the object that you are visualizing, while the eyepiece actually analyzes this image that is produced by the objective lens, and it produces a virtual and magnified image, virtual or erect image in this case. So I think this answer is clear. Anyone need any particular explanation or any other doubt? It's actually like easy if you understand the line diagram or lens diagram and all. So I think I will just move on to the next question then. So coming on to question number 12. This basically I have given you the answer. The image produced by objective lens is which one, and that of the eyepiece is which one? First one is real real, second one is real virtual, virtual real, or virtual virtual. Which might be the answer? Okay, I am getting an option real and virtual. I am also getting an option 3. Any other option? Option number 2 I am also getting. Okay, so the answer to this question is option number 2, real and virtual image. So image produced by the objective is real and inverted, and that too by an eyepiece is virtual and erect. So basically I will just you can see that I will just give you an explanation once more. So if you have if the objective lens is looking at an object, then the light rays from the objective will enter into a lens. So it will move, this particular light ray will move directly, and then the second light ray that moves hit the side of the lens will get converged into a position. So then the convergent point actually creates an image there. So if you compare these two images, this object as compared to the image that is produced by the objective lens, right, you can see that it is inverted. So this moves towards here and this moves towards here, right? So it's actually an inverted image in this case. So and also it's actually something that, because the image is formed by convergence of light rays, right, so it converges here, it's actually called as a real image if it is converging. So it's real as well as inverted image that is produced by the objective lens. So using this particular image that is formed by the objective lens, the eyepiece actually provides a magnified image of that image itself. So if you continue on this light rays, you can see that it hits this particular eye lens rays, eyepiece there. So if you move on to here, you can see that it will actually does not converge, it moves on towards infinity. It's like a triangular cone there, right? So basically it's a something that cannot be converged there. So in order to extrapolate our information, then what we are doing is that from the eye you can see that you can actually make a line diagram up to this particular point, and from this particular initial image you can see that this particular image that is created, or final image that is created, is much more enlarged as compared to this object's image, right? So basically as it moves into infinity like a cone of light, you can see that it's actually a virtual image, virtual enlarged image, and it is an erect image. Basically it cannot be extrapolated into a single point, as it diverges out into infinity and it reaches our eye point. So basically the image produced by objective lens is actually real, and that by the eyepiece is actually virtual in nature, real and inverted, while eyepiece is virtual and erect. So I think that explanation is clear. Anyone has any doubt? Okay, so I think that's clear. I will just move on to the next question then. Question number 13. Osmolality of human plasma is 150 milliosmoles per kilogram, 190 milliosmoles per kilogram, 290 milliosmoles per kilogram, or 400 milliosmoles per kilogram. Okay, I'm getting option number 3. Yeah, so you are right. So option is actually 290 milliosmoles per rate. So it actually fluctuates somewhere in between. So basically, if you look into the normal osmolality of human plasma, is about 275 to 295 milliosmoles per kilogram, and basically yeah, that's the answer basically, because if you look into osmolality and all, basically it's a kind of a colligative property, right? So basically it depends on the number of solutes that are present within the cell. If you look in, compare a cerebral final fluid or a CSF with that of human plasma, it will be different. So each and every cell type that you are growing, which is obviously different in nature, has a different kind of osmolality. So whenever you are what you say, whenever you are taking a cell type and growing it in a media, you have to check the osmolality of the media first before you are putting in a cell culture, because chances are that if the osmolality doesn't coincide with the osmolality of the cell type you are growing with, or the conditions that the cell type needs to grow with, then chances are that the cell culture proliferation or any other phenotype that might be seeing might not work at all. So basically you don't know one type basically. So when you are having a basic understanding of cell culture and all, you should know the osmolality of the cell type you are growing, and what kind of cell culture you should provide, and what should be its osmolality. so that the cell culture can proliferate rapidly without any interference from the media or any other kind of buffer that you are using. So it actually lies in between 275 to 295, and based on this question, the option number 3 is the right answer. So that the osmolality of the human plasma is 290 milliosmoles per kilogram. So basically, this is the unit that you have to recognize. Whenever you are saying a value, you should know what the unit is, and you should describe what the unit is also. So anyone has any doubt regarding this? Okay, so basically, if you are looking into osmolality and all, basically there's a instrument called as osmometer basically. So basically, osmolality is kind of a colligative property, right? So based on some of the factors that I have earlier discussed, like reduction in vapor pressure, so and the what you say, depression in freezing point and all. So basically, this osmolality is dependent on all these factors basically. So whenever osmolality is dependent on the number of solute particles also, because it is a kind of a colligative property, right? So whenever the number of solute, actually the number of solute, ratio of number of solute to that of the solvent molecule within that particular solution, that is the cell culture of the buffer, actually dictates what the osmolality is, and based on that you need to discuss on the what are cell culture like media you are using or the buffer you should be using basically. So it's actually determined by an instrument called as an osmometer, and this osmometer works on the principle of this colligative property, changes in colligative properties like reduction in vapor pressure, depression in freezing point, elevation in boiling point, etc., and changes in this osmotic potential or pressure there, the movement of one solution from one point to the other solution, which is separated by semi-permeable membrane there. And I think that's clear basically. So I think that's clear, right, basically. Okay. So anyone has any other doubt? Okay, so I think it's clear. So I will just move on to yeah. So I will just explain. I actually forgot about it. Sorry for that. So basically, this is an osmometer there. So you can buy different brands and all. So osmometer is a specialized device used to measure the osmotic strength of solid solutions, colloid or compounds. So it is particularly important in clinical laboratory for determining the concentration of particles in a solution. That's what I said, right? Osmolar concentration is called osmolar concentration, the number of solutes that are present in a solvent, right? So basically it has two different units, milliosmoles per kilogram, millimolar per kilogram, or for osmolarity, or millimolar per liter, or for directly for osmolarity you can also use this unit, that is milliosmoles per kilogram. So these particular like factors like freezing point, depression, vapor pressure, like changes in vapor pressure, or that decrease in vapor pressure, or that membrane osmometer, like how to detect the changes in the osmotic pressure by movement of solution from one solution to the other, and also the elevation in boiling point. These kind of factors are determined by this osmotic meter, and based on that it gives a value, like you can see that 290 is written there. 290 milliosmoles per kilogram is basically the value of this particular solvent that they are used to analyze this particular compound that is kept in this instrument. I think so that they are using that, or just to show for any promotion and all. So I think this is clear, and I will just move on to the next slide then. So coming to question number 14, which microscope is used to study samples from below? You know the answer to this question. First one is compound, phase contrast, confocal, or inverter, which might be answer from the below. Yeah, I'm getting option number 4, inverter. Option number 4 again I'm getting. Any other option? Yeah, you are right. So the answer to that question is option number 4, that is the inverted microscope. So I am not going into much detailed factor in this case, because I have already discussed with you, because the whenever you look into inverter microscope, right? So there's a light source at the top, then there is a sample that is kept there. So light actually falls into a sample, then it moves out, because the in this case you have to see that the sample or the cow slip that you are using is either plastic or glass, and based on the refractive index of that particular substance, then you have to choose a microscope and all, as I have discussed earlier. And then it comes out, then it moves into the objective lens, right? So there's a light source there, condenser there, diaphragm is there, and then light enters into, hits the specimen there, it moves out into, it gets reached the objective lens. From the objective lens, it moves up with the presence of mirrors or anything like that, or any kind of lens or anything like that, then it moves into eyepiece. So you can directly visualize it by your naked eye, or basically what you say, you can either record it in a particular computer. Okay, so I am getting a question, can you explain the principle of inverter microscope? Okay, so I think I will just move on to the previous slide. Yeah, so these are just type of a I don't have the exact principle of how light rays actually moves through a microscope, but I can just give a brief explanation how light travels and reaches the eyepiece through objective basically. So basically, if you look into a upright microscope as well as inverter microscope, upright microscope is basically used when the if the object is actually, or sorry, the sample specimens or the slide that you are using is actually opaque or translucent in nature, because the light rays then have to pass through the light actually comes out of the top of the microscope, hits the microscope, and then it moves into the objective and then into your eyepiece. In case of inverted microscope, you cannot use any opaque or any translucent like sample stage or anything like that, because the light actually comes from here, then it enters objective, then it passes through the sample that is kept there, right? So there is a light there, objective lens is there. From the objective lens, sorry, that was a mistake. The light source actually kept at the top, right? So light actually moves through the sample that is kept there, then it reaches the objective. So the image is formed with the objective in between here, as because each and every, like the combo microscope has similar principle of action and all. So it moves through this objective, and somehow through reflection or any other process, it can move through the eyepiece and all. So it reaches the eyepiece, so the light actually travels from here, it hits the sample specimen that is kept here, then after the light has been gathered, then it moves through objective, and then it moves into the eyepiece or to the camera, that depending on what you want to do, it actually produces a particular image. So the difference between the upright image and inverted image is that basically the light is actually coming up from the bottom in case of upright image, and in case of inverted image it actually comes from the top. Because of the sometimes the sample specimen that you are using is might be opaque or translucent in nature, so upright microscope is used there. If it is not opaque or translucent, basically in case of inverted image in microscope we cannot use that opaque or translucent image, because the image actually comes the light actually comes from the top, right? So if it is opaque, then it will get blocked here, it won't pass through objective. So basically yeah, these are the two principles in case of inverted microscope and upright microscope. The exact principle I haven't shown anything here, but if you look into the principle of how light travels through a microscope, I think wait a minute, this will give you an idea. Basically you can see that how light travels through a microscope, right? So basically like it's similar in each case, both the upright as well as the inverted microscope. So I hope this is clear. Yeah. So basically, in case of if you look into a cell culture and all, right, you are using this particular kind of transparent tubes and all, which is not opaque, and in case of microscope. And also, if you look into the objective lens, basically the objective is produced. Sorry, I will just move on to the previous slide. So the objective is kept here, right, as compared to an upright microscope which is present here. So objective lens will be much closer to the sample basically. So the box will be kept here and the sample will be present at the bottom, right? So compare that with here. In case of upright microscope, the box is kept there, sample will be at the bottom. So you can see that if you look into a distance that the objective covers in case of to the sample that you are looking in inverted microscope as compared to upright microscope, the objective will be much closer in case of inverted microscope as compared to an upright microscope, because if you keep the sample here, the first barrier will be at the the top, right? There is no cell growing there. The cell will be growing at the bottom of the cell culture, right, which is kept there. So in this case you can see that the objective lens will be much closer to the cell culture plate in case of inverted microscope as compared to an upright microscope. But you have to keep in mind that you have to use the cell culture like plate which is obviously transparent in nature. So I think and that will give you much resolution or much higher clear image as compared to that upright microscope. So I think that's clear. Okay, so anyone has any other doubt? Okay, so I think that was a good question. Thanks for that question. So I will just move on to the next question, then question number 15. So hydrophobic substrate can be used for which of these cultures? Anchorage dependent cell culture, anchorage independent cell culture, monolayer, or histotypic cell culture? So I am getting option number 2, anchorage independent cell culture. Any other answer? If you attended the previous section, week 1 section and all, I think you will know the answer to this question. It's actually, if you know the basic principle, what these questions are, you will know the answer. So I will just give you answer. If you are right, answer is anchorage dependent cell culture, because like a hydrophobic surface can be used for which of these culture, because basically if you look into an anchorage dependent cell culture, right, basically anchorage dependent cell culture are those culture cells that proliferate or divide in the presence of an extracellular matrix, right? Basically in the presence, if you look into a cell culture and all, there will be different extracellular matrix like collagen is there, poly-L lysine, poly-L arginine, several are there, right? These provide an extracellular coating at the surface, and these are basically like hydrophilic in nature, right? So that basically when you are incubating a cell culture in there, these cells that are anchorage dependent actually binds to this culture, and then only then after binding to this extracellular matrix or an artificial or a natural extracellular matrix, then they will start to divide and grow and also proliferate and then show their phenotype, whatever is shown basically in that medium. So basically these anchorage dependent cell, if you look into the first option, anchorage dependent cell actually require these particular extracellular matrix within the culture plate, which can be collagen or poly-L lysine, arginine, etc., whatever it is, where it is artificial or natural, like it depends. So these are basically, as I said, hydrophilic in nature. So if you are providing an because the if you look into the interactions that are taking place between this anchorage dependent cell as well as the extracellular matrix is actually protein-protein interaction. So protein-protein interactions are mostly hydrophilic in nature, right? Not hydrophobic. So in this case, if you are providing a hydrophobic environment into anchorage dependent cell culture, it won't attach to that particular surface because it is not the surface that you are providing. Sometimes you can have to modify this hydrophobic surface so that anchorage dependent cell culture can bind to it and then divide and grow. That's a different thing. But if you are providing just an hydrophobic surface, it cannot bind to that particular surface, neither can it undergo proliferation. So it needs a hydrophilic extracellular matrix for attachment so that it can grow, because these are all protein-protein interaction, example integrin fibronate interaction, integrin proteoglycan interaction, whatever it is, women interaction. So whatever it is. So anchorage independent cell actually are provided within that suspension culture, right? Because it doesn't attach to extracellular matrix, it's actually suspended within that culture medium. So it doesn't require this particular hydrophobic, whatever the substrate is, it can grow because it doesn't require attachment to extracellular matrix for its proliferation or growth. Monolayer culture also similar. It is similar. These are anchorage dependent cell culture. However, it grows as a film it doesn't divide at the top and all, it just grows as a single layer there, and it depends on the extracellular matrix which is hydrophilic in nature. Histotypic culture is also consists of a kinds of a multi-layer culture. It is also anchorage dependent in nature, like 3D spheroids and all etc. So it requires this particular like extracellular matrix to attach itself to and then undergo cell division as well as proliferation. So in this case, it cannot attach to it cannot attach itself to hydrophobic surface because it requires a hydrophilic surface for protein-protein interaction and the presence of an extracellular matrix. So I think this answer is clear. Anyone has any doubt? Okay. So I think this is quite clear because if you know the basic, you will get the answer to this question, right? So coming to question number 15. Dash of a microscope is a dimensionless number, wavelength of an incident light on lens, optical resolution, numerical aperture or focal length of the lens. Which might be the answer? Okay, numerical aperture and get any other answer? Okay, so I think numerical aperture is the answer. So option number 3 is the answer. You're right. Numerical aperture of the microscope is a dimensional number because it's actually a multiplication factor of this particular. This is what refractive index, right? Refractive index. That is a ratio of a medium number 1 to the ratio of refractive index of medium number 1 to the ratio of to the refractive index of medium number 2, that is from the specimen to the objective lens that you are looking at. So that is n, and into sin theta. Sin theta is basically an angle, right? It's actually, you know, that is a unitless compound. So basically determine numerical aperture, I'm just repeating, it is a factor or a parameter that quantifies the ability of a microscope objective to gather light and resolve fine specimen details at a fixed object distance. So basically, you know, equation numerical aperture is a ratio, the refractive index into sin theta. Basically, these two are unitless factors. So you are when you are multiplying a unitless factor with another unitless factor, it always yields a unitless factor. So it is kind of a dimensional because of the absence of this unit. It's actually a dimensionless factor. So I think this is quite clear. Anyone has any doubt? Okay, so I think it's clear. So I will just move on to the next question. Yeah. So I think you know the answer. Osmometer works by dash of a medium, depressing freezing point, depression of freezing point, increasing temperature, increasing pH, or decreasing temperature, which might be the answer. This we have discussed basically. There are several answers, but based on this question, what is the answer? Okay, option number 1. I'm getting depression of freezing point. Any other answer? Okay, again I am getting option number 1. Okay, option number 1. So, answer to this question is right, depression of freezing point. So basically it works on the basis of the colligative properties, right? Colligative properties are dependent on the number of solutes that are present within that solvent space, right? So basically if you are increasing the number of solutes or adding the solute into a homogeneous solution, the homogeneity gets decreased. So in that case, chances are that when you are putting it in a low temperature, the freezing point also gets changed accordingly. Most probably it will get depressed because decreased because of the presence of the solute there. So the answer to this question is osmometers work by depressing freezing point, and it is dependent on the colligative properties and all. So anyone has any doubt? Okay, so I will move on to the next question. Polystyrene has a hydrophobic surface that dash skeletal muscle cell growth. So first option is promote, allows late, disallow, or allows faster. Which may be the answer? Disallow I am getting. Any other option? Option number 3 I am getting. Yeah, so you are right basically. So it does allow polystyrene has a hydrophobic surface that disallows skeletal muscle growth. Because you know that skeletal muscles are like anchorage dependent cell lines, right? Because for whenever you are providing incubating it into a cell culture and all, it needs to bind itself to an extracellular matrix that are produced within the cell culture plate, right? So upon when it binds to that extracellular matrix protein and all, then it undergoes cell signal cascades within it, which tells it to like, okay, you need to proliferate and divide now and show whatever you need to show there. So without the presence of this extracellular matrix, it just remains in suspension, it doesn't grow at all. So hydrophobic surface does not promote this. You need a hydrophilic surface there in the presence of like natural polymers like collagen or organic polymers, artificial polymers like polyl lysine or polyl arginine there, because in order to promote the binding to extracellular matrix, protein interactions with the integrin of the cell skeletal muscles need to take place, so that it binds to it and then it starts to proliferate as well as grow. So in case of hydrophobic surface, which is not a hydrophilic surface, it needs a hydrophilic surface to grow for this attachment to place. If you are providing hydrophobic surface, there is no cell attachment there. So even though you are providing it, it won't bind to that and it does not grow basically. So polystyrene has a hydrophobic surface that disallow skeletal muscle growth. Skeletal muscle cell growth. So does anyone have any doubt regarding this? Okay, so I think this is quite clear based on the previous question that I just made. So yeah, it's additional information I have just told that, so I'm not repeating it. So question number 90. What can be used to know if a surface is hydrophilic or a hydrophobic? Temperature measurement, pH measurement, contact angle measurement, or X-ray diffraction, which may be the answer. Yeah, I'm getting option number 3. Anyone has any other answer? Yeah, basically answer is contact angle measurement. So if you look into a contact angle measurement, what you need to do is that you have to put a drop of some particular solution onto the surface that you are analyzing. Suppose that this is the polystyrene surface or any other collagen surface there. So you are putting a drop of any kind of solution there. So basically, if you are looking into a hydrophilic surface, right, hydrophilic solution or a kind of water, like if you are putting a water droplet at the top, you can see that if water is basically hydrophilic in nature, right? So if you are putting a drop at the top of a hydrophilic surface, chances are that the cohesion forces that are taking place within this surface will be more, and then the water will be spread out if it is put up in a hydrophilic surface. So the drop will more evenly spread out, and the angle of contact, that if you look into the angle of contact between this surface to the drop surface, you can see that if the angle of contact is less than 90, then it will be hydrophilic in nature in case of water. But if you put it into a hydrophobic surface, say like polystyrene, then it will form a more rounder drop there. So if you are comparing the angle between this contact angle between this water molecule surface with that of the hydrophobic surface, you can see that the angle will be much higher, larger as compared to that of more than 90 degrees as compared to that of this particular hydrophobic surface, where the water is put in case of hydrophilic surface, it spreads out. In case of this hydrophobic surface, it wouldn't spread out. So there is a difference in angle there. Based on the difference in angle that has been generated there, you can say that whether that particular surface that you are analyzing or examining is actually hydrophobic or hydrophilic in nature. And an instrument called as goniometer or a contact angle goniometer is actually used. So basically, if you are putting, there's a needle there, you are putting a sample there, and there's a surface that you are basically examining there. So the needle comes down, it produces a drop, and it slowly drops this particular drop onto a surface, or the hydrophobic or hydrophilic whatever surface, the surface which is under examination there, and then it takes an image there. So it records this particular image, how the drop is formed there. Then based on some application or mathematical calculation there, it then based on the angle that it is getting or analyzing, then it provides that whether the surface is hydrophobic or hydrophilic there. and the solution is usually colored in nature so that the angle can be measured there using other applications and all. So this is how contact angle measurement actually take place, and it is measured by a instrument called as goniometer. So anyone has any doubt? Okay, so blood has hydrophobic and hydrophilic surface. So I think it's hydrophilic in nature, right? I'm not that sure, but I need to check that out. I think it's hydrophilic in nature. So can I just check it out and then answer that question? Okay, so thank you for that. So I will. Anyone has any other doubt? Okay, so I will move on to the final question then. Question number 20. So which of these are anchorage-independent cell culture? Hematopoietic cell culture, cardiac cell culture, skeletal muscle cell, or neuronal cell culture? Which might be the answer? Okay, option number 1, hematopoietic cell culture. Basically, basically you know the basics, right? So it's basically hematopoietic cell culture, which is basically related to blood, right? So blood generation and all these are cells that give rise to other blood cells and all. So basically it is present within our blood in suspension, right? It doesn't attach to other cells and all. So basically it does not require any extracellular matrix for attachment. So basically it can grow in a cell suspension and all without the presence of any extracellular matrix or whatsoever you are providing. So that's why it's called as anchorage independent cell culture, because it doesn't require any extracellular matrix for its growth and proliferation, as compared to other like cardiac cell culture or heart cell culture, skeletal muscle cells, or neuronal or nervous system, whatever it is. So I think that's clear. So anyone has any doubt regarding this question? Okay, so I think we can end this section based on this particular question. I think explanation is also clear. So anyone has any doubt, you can ask right now. Anything from the whole topic that we have covered today, or anything else like that? Okay, so I think everything is clear. Basically, some of the doubts were asked, also those were also good doubts. So anyone has any doubt, you can also ask in the next slide also. So I think I will just end the presentation here for today. So thank you everybody for attending the class. I hope that for the next session you also attend the class also. So thank you. Thank you.